In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory. My name is Som Shuvra, and I'm joined by my colleague Janine. Our technical information services team serves the research community with in vivo research education questions and offers answers to all your technical queries.
For any of you watching live, you can use the comment section of the video to share with us where you're viewing from and any commentary you have on today's subject. So in 2 days, on April 7th, our colleague, Dr. Brian Soper, will present a webinar on in vivo preclinical efficacy and safety of CAR-T, NK, and myeloid therapies. So today, let's talk fighting cancer with CAR-T-based therapies. So to help set stage for that webinar, our JTT today, our 53rd episode, we're going to review some of the introductory aspects related to how chimeric antigen receptor, or CAR-based therapies, are being used to fight cancer. Som and I here are not involved in developing any of these therapies. We're just both very enthusiastic about the subject because of our previous PhD and postdoctoral training in oncology. And we've just been reading up on the subject more recently. So most of what work has been done in CAR-T, or T cells. So that's going to be our focus today. But there are also similar strategies that are being employed for other immune cells like NK cells and myeloid cells. So Som, what are the CAR molecules and how are they being used to fight cancer?
Great question, Janine, and this is a great question to start the conversation today. Well, we know cancer cells are smart, and they'll take every possible way to counter evade the immune system. Hence, training these inherent immune cells is perhaps the most smartest way to reduce this evasion and also effectively fight cancer. In CAR-T therapies, for example, first, T cells are removed from a patient and then engineered to express a tumor-specific CAR using gene transfer technology. Finally, the genetically engineered cells are put back into the patient. What makes CAR special is that it expresses a membrane-bound protein that allows the cells to recognize their target antigen on the tumor by a single-chain variable fragment, which we also know in short for SVFP binding, mimicking the way antibodies typically bind to the targets. The SPV portion is linked to an intracellular domain with components that activate the T cell with bound to the target, which results in very effective killing of the cancer cells. This therapy actually has numerous advantages, such as efficiency of T cell expansion, avoidance of tissue rejection when a patient provides their own T cells, and the possible prevention of tumor recurrence using reinfused CAR T cells. Would you like anything to add to that, Jenny?
Sure. I can talk about the different sources of T cells, and actually Mohan, who's joining us on LinkedIn, hi Mohan, is asking us about the differences between allogeneic and autologous benefits. And so I'm actually going to talk about that now. And just to remind all of you watching that you can submit your commentary just as Mohan has done. So Som, you just mentioned you can avoid tissue rejection if a patient is providing their own T cells, and this is called autologous CAR-T therapies. Where the patient donates essentially their own cells back to themselves. So right now, the USDA has approved cellular and gene therapy products, and it includes 7 autologous CAR-T therapies. 4 of those are for B-cell lymphomas, 2 are for multiple myeloma, and 1 for prostate cancer. So in contrast, when those T cells are actually coming from a donor population, this is called allogeneic therapy. And currently, the primary FDA-approved allogeneic therapies from that same list that I just talked about are just cord blood, which are coming from donations from cord blood. And those are a really large source of hematopoietic stem cells, and they're typically used in stem cell transplants that can happen after something like myeloablation. So myeloablation is another therapy that's used to address some of those same heme malignancies that I just talked about, leukemias, lymphomas, and multiple myelomas, but they're not targeted to the cancer cells. They're things like chemotherapy and irradiation. And so in that process, they're also going to damage a lot of normal bone marrow. And so those allogeneic therapies of cord blood donations would be needed to provide those normal stem cells back into the patient to develop normal blood cells. So it would actually be really advantageous to develop allogeneic CAR therapies that would then address the tumor specificity concern that myeloablation would not be able to address. But also allogeneic CAR T would also allow you to generate a library of ready-to-go CAR Ts. And that would all be coming from healthy donors where there would be a larger population to draw on and a lot more material to work with. So that would allow a lot more scalability of the therapy, as well as increasing the speed and the variety of the different treatment options that are available to those patients. And then lastly, allogeneic CAR T would actually be a lot easier on the patient receiving that therapy because autologous CAR T therapy is actually a pretty lengthy process. And the patients that are undergoing autologous CAR T actually have to be healthy enough such that they can go without any kind of treatments for a few weeks leading up to their leukapheresis procedure. And then they have to wait for a company to then take those cells, engineer them, and then return them back and reinfuse them. So that can actually be a pretty challenging process for those patients receiving that therapy to undergo. And allogeneic would avoid some of that. So allogeneic CAR T, of course, gets a lot more complex because, as you said, you're using donor cells now, and the autologous would be, you wouldn't have the problems of rejection. So when you do allogeneic CAR T, there have to be methods to prevent those donor T cells from then attacking the patient's normal tissues, which would be a graft versus host disease. And there also has to be a method that prevents the patient from eliminating those donor cells in a process called allorejection. So there's a lot of kind of pre-treatment therapies that have to be done to account for an allogeneic CAR T. So, can you tell me about the different ways CAR T molecules are being designed? From what I understand, we're kind of, in the cancer field, are always looking for tumor-specific antigens, and that's kind of basic monoclonal antibody design principles. So I think the most universal improvements in CAR technology would be on the T cell side of things. So I've heard a lot about different generations of CAR Ts and even trucks. What are those?
Yeah, great question. There are many generations of these constructs to highlight, actually, and there have been incremental improvements made in these CAR designs that have been that resulted in a lot of those different generations. For example, the first generation construct actually harbored a basic structure with one signaling region with an additional co-stimulatory domain, such as CD28, for example, that was being included in the second generation, and then incorporation of multiple additional co-stimulatory domains being added to even after that in the third generation and on. More recently, a fourth generation CAR, which are known as the armored CARs or trucks, as you said. Trucks actually stands for T cells redirected for universal cytokine killing, is being tested upon. The fourth generation CAR T is actually designed to shape the tumor microenvironment by the inducible release of transgenic immune modifiers, for example, IL-1/2, let's say, which augments T cell activation, attracts and activates innate immune cells to eliminate antigen negative cancer cells in the targeted lesion. Having said that, I want to give you an example, Janine, today. I'd like to mention the JACR017 and the JACR015, two agents by a company called Juno Therapeutics. Both of these, JACR015 and JACR017, are directed against CD19, but the agents have very, very different constructs. I'll tell you some of those. For example, in JACR015, the cells they used are composed of CD3 plus enriched peripheral blood mononuclear cells, whereas JACR017 is actually built on a fixed ratio of CD4 and CD8 positive T lymphocytes. Each agent is also engineered differently with different viral vector, namely retroviral for JACR015 and lentiviral for JACR017. The binding domain for JACR015 and JACR017 is also different. For 015, it is SJ25C1, and the co-stimulatory domain is CD28, whereas JACR017 has a binding domain of FMC63 and a co-stimulatory domain of 41BB. In addition to some of these differences that I just mentioned to you, JACR017 also contains an additional ablative technology to provide better control of proliferation and survival of the engineered T cells, which is actually not included in the other one, which is JCAR015. This ablative technology, it's just a truncated form of the human epidermal growth factor receptor, or the EGFRT as we know it, allows for the rapid killing of the CAR T cells using any epidermal growth factor receptor inhibitor, let's say cetuximab or anything like that, that is can be added if needed. Juno also mentions that the addition of the truncated EGF receptor may have some kind of immunostimulant properties to it.
So there's a lot of thought that goes into designing these molecules. I mean, you can be really ingenious with all the different co-stimulation if you know kind of basic T cell biology, but then kind of all the other engineered protections or enhancements to these CARs is really, really ingenious. So it's nice to kind of understand the rationale behind how people develop these therapies. Just looking at our comments, Grace is asking what kind of risks you look at besides, tissue rejection of the patient in CAR Ts. And since you brought up Juno Therapeutics, Solom, it's probably a good time to talk about that. So I started reading about CAR T molecules in early 2016, and I was actually just reading about this because I wanted to write a blog on CAR T molecules. But that was also when JCAR015 and JCAR017 were being developed, and the FDA was naming this a breakthrough therapy at that time because there was a huge promise for efficacy there. I wrote the blog article, and then later that year, in the middle of 2016, Juno had to halt phase 2 clinical trials of JCAR015 because 5 people on that study died of neurotoxicity. So Grace, you're bringing up another good point about risks to health. So in this study, the clinical trial, that medical directors said they described a number of factors that were associated with those deaths. One of them was an increased level of the cytokine IL-15. IL-15 is a cytokine that can improve T cell activation and function, which you would think is a great thing for this type of therapy, but it can also get out of balance. And I think that's what they were starting to see, and what is commonly seen in a lot of CAR T is this out of balance and this over activation of cytokine release. So the other thing that they noticed in JCAR015 was those patients had a really rapid expansion of CAR T cells, including very high levels of cytotoxic CD8 positive T cells. And those cytotoxic T cells can secrete inflammatory cytokines like IL-2 and TNF alpha, and those can go on then to create a lot of damage in the blood-brain barrier. So that was their mechanism for developing the cerebral edema and the neurotoxicity that they saw in those 5 patients. It was also, their experience as well as others in the field later about the different co-stimulatory molecules that were employed. So some you mentioned CD28 and 4-1BB. So Juno had their 2 different molecules, 015 and 017, and one of the differences that you mentioned was the differences in the co-stimulatory molecules. So JCAR015 had the CD28 version of the co-stimulatory molecule, whereas JCAR017 had the 4-1BB. And both Juno and others in the field observed that the CD28 containing versions really expand quite rapidly in vivo, whereas the 4-1BB versions tended to contribute to a longer persistence in the blood. So Juno was actually able to move forward with the JCAR017 because of they were able to manage those toxicities, and they didn't have that rapid expansion that they experienced with the JCAR105. But it's not as simple as that. So it's not just a matter of switching out the different co-stimulatory molecules, because actually at the same time as Juno, both Novartis and Kite Pharma were also developing CAR T, autologous CAR Ts for CD19, and actually both of those went on to get approved. And Novartis and Kite, I can't remember which one, but they were also using CD28, and one of them was also using 4-1BB, and both of those ended up getting approved. So it's not just the that co-stimulatory molecule that determined everything. It was kind of the design of a lot of different things that contribute to the success of those drugs during phase 2 and beyond. And it's actually interesting because both Kite and Novartis also experienced cytokine release syndrome as well as neurotoxicity in phase 2. So those weren't the only factors that contributed to the loss of the first Juno molecule. And so as we've seen, as more CAR-T has been going into clinical trials, that CRS and neurotoxicity is actually a pretty normal expectation of a lot of those types of therapies, just based on the type of therapy and how they work. So to address this, there does have to be a pretty good understanding of the CAR-T and its potential to induce CRS, or the extreme form, which is called cytokine storm. And then there also has to be a very good understanding of who in the patient population should be allowed to enroll, which then eventually informs who can actually be treated in the future. And there's a lot of different things that clinicians can do to kind of precondition their patients before they undergo this very efficacious therapy in order to kind of prevent CRS and neurotoxicities. And so they might have to do some kind of preconditioning therapies to get them ready for that, and then reduce the risk of those toxicities.
Great examples, Janine, and this is very important to know because one part is doing the novel aspects of it, but the toxicities is absolutely something that needs to be considered. And this is exactly where the upcoming JAX webinar by Dr. Brian Soper on April 7 comes into play. It might be an excellent resource for all of you to have more information on these associated toxicities. In this webinar, Dr. Soper looks to cover some of these very limitations you were mentioning, Janine, including durability of response, lack of solid tumor efficacy, cytokine release mediated toxicities, and so on. Please, all of you who are viewing this today, remember to participate in this webinar if you'd like to learn more. If you have not already registered, you can also do so by using the link we have provided in today's JTT resources available below the video that you are viewing.
So there's a question from Arad. I'm sorry if I mispronounced your name. His question is, and I don't know the answer to this, why have researchers decided to focus on NK and dendritic cells in addition to CAR-T? You've been reading a little bit about those. Do you have an idea?
Yeah, that's a great question. Thanks for that. So it might be a lot of different myriad of reasons why people are trying to develop those therapies. It's not just one reason why it's bad or good. It's just a different approach, to be honest, which I feel depends on what you're trying to study in terms of the disease, and also the different approaches. This is a way more a specific approach from each one of those avenues as you go, like CAR-NKs or CAR-dendritic cells. And each one of those approaches will perhaps have their own pros and cons, right? So it depends on what you're trying to achieve and where you want to take your study design, I guess. That's probably the rationale, and it's just making more advances than we ever imagined that this field would be going towards. So it's really good that it's going there, but one thing to know is always keeping an eye out. I have seen some of those papers, and they are all built upon the research that have been done on CAR-T, and considerations, limitations, and advantages are some things that are always at the hem of all those studies. So it's definitely one of the alternative ways, I would say, that why people are using one of those designs to approach treating cancer.
Hey, while you were talking, I kind of thought a little bit more, so thanks for buying me some time to think. I was thinking about Arad's question too, because T cells, essentially what we're doing is we're, in CAR-T, you're stimulating, you're taking the antigen specificity of the antibody kind of outside, right, to identify the cancer cells, and then you're taking advantage of T cell signaling to stimulate T cell specific functions that T cells are really good at. And so NK and dendritic cells also have their niche in the immune system, and I'm not as good at the immunology here, but they're going to have different effector functions. So I imagine that NK and dendritic cells are being used depending on the biology of those cells and how they fit into the type of tumor. And what you're trying, or I guess what those therapies are groups developing them are trying to get in terms of effector function of those cells. So NK cells are going to be direct killing, and so maybe there's ability for a direct killing that maybe is going to avoid some of the T cell functions that maybe are more problematic. I don't have a good handle on that, but I imagine it has something to do with the biology of what those cells do and how that might be more applicable to the type of tumor that they're trying to address.
Right.
All right, I think we're out of time. We were going to talk a little bit about solid tumors. I think we can like really quickly talk about solid tumors, right? So, you know, there's a most of the therapies have been directed towards heme malignancies, but there's got to be some modifications for solid tumors. And I think the reason why heme malignancies were kind of the first target for CAR T was just their accessibility. You're going to inject these CARs directly into the bloodstream, and how convenient that these you know, heme malignancies are in that same location. So I think that has to do a lot with the architecture. Whereas solid tumors are going to be a lot more complex. It's going to be harder to get to them because of the location where they are, how much extracellular matrix is present, how much vascularization is possible. But I think some of the modifications for solid tumors are actually, people are looking to directly inject CAR Ts into solid tumors. What else have you read, Som?
Yeah, that's a good point. I mean, each tumor brings its own niche challenges, right? So solid tumors brings its own as usual. So solid tumors, you know, they do not have the same mix of antigens perhaps on the surface, for which a CAR regimen can most likely miss some of those that it needs to target. The tumor microenvironment is very important to consider when working with solid tumors, because solid tumors are known to generate immune suppressing agents, let's say PD-L1 on its surface, which is not going to be the challenge in liquid tumors. Another aspect, a physical aspect that comes with solid tumors, Janine, is solid tumors are very dense and they have several layers thick. Infiltration itself is a challenge for these immune cells to go into the deeper layers. And perhaps the cells that reach those layers, other conditions like tumor hypoxia, for example, can be present there that alters or creates issues for the survival of those cells. So absolutely, I agree with you. Solid tumors brings its own challenges, but it's all about, you know, engineering around that.
All right, everybody. Thanks for your participation. It's all the time we have for this week. Really enjoyed the commentary from people who joined us live. So I hope you guys keep coming back and found this valuable. Just to close out, there are 2 links in our video description. We've only mentioned this like 4 times. There's a registration link to the webinar in 2 days. And then there's also a blog article, not the one that I wrote, one of my colleagues wrote, on CAR T. And if you like reading, that's another method for you to read about CAR T and what we've been describing today. And our next JAX Tech Talk is scheduled for April 19th. It's called Let's Talk Het3 Mice for Testing Anti-Aging Compounds. You can follow JAX Tech Talk on LinkedIn, as so many of you have today. And then you're going to be notified anytime we have a new episode, or you can subscribe to a playlist that we have on YouTube. This is Janine and Som saying stay healthy, stay safe, and stay excited about research. Bye, everybody.
Bye-bye.